Hello everyone, my name is Alessandro Busetti and I am one of the field application scientists for Metabolon's international sales team. And today I decided to share a beautiful case study highlighting how not all viruses are bad, and in fact how some of these can be harnessed for their incredible biotechnological potential, and most of all, of course, how Metabolon's Metabolomics platforms proved invaluable for the study and provided invaluable insights which will eventually help to unlock this incredible biotechnological potential. This is a study about a very specific type of virus called a bacteriophage, or phage in short, which is a type of virus which infects and parasitizes bacterial cells. It was published in Cell Host and Microbe in 2019, and the first author is Brian B. Hsu. The bacteriophages in this study are so-called virulent phages, as they follow the so-called lytic cycle of viral replication, which results in the destruction of the infected cell and its membrane. Now, after the bacteriophage has injected its genetic material inside the bacterial cell, it hijacks the cell's replication and translation mechanisms and uses them to make more viruses. Then specific viral proteins cause the lysis of the bacterial cell and the release of the new viruses or virions in the surrounding environment, where this infection cycle can begin again. One last concept before I jump into the case study, and that is host range. Now, each phage has its own particular host range, which is the range of bacteria that they can infect. While some phages can only infect one or a few bacterial strains, other phages can infect many species or even bacteria from different phyla. But generally speaking, phages tend to be highly species and strain specific. And this is a very important characteristic and attribute, especially from the point of view of this study. Yes, and finally, let's jump into this beautiful case study. So, the human microbiome is comprised of densely colonizing microorganisms, including bacteria and bacteriophages, which are in dynamic interaction with each other and with their mammalian host. Now, the microbiome is known to benefit human health via a myriad of mechanisms, including metabolic cross-feeding and immune system priming. Conversely, an imbalanced or depleted or dysbiotic microbiome can be harmful and has been associated to a number of diseases and disease states, including poor nutrient utilization, gastrointestinal disease, liver disease, heart disease, central nervous system diseases such as neurodegeneration. And so as we gain a greater understanding of the role of the gut microbiome in human health and disease, the leading question becomes, what factors in the gut influence the microbes that influence us? And how can we leverage this knowledge towards treatments that manipulate the microbiome's composition? Now, current approaches to modulating the gut microbiome include dietary changes like the use of prebiotics or the use of probiotics, or even the use of antibiotics. But these strategies cause broad and potentially long-lasting perturbations, not to mention antimicrobial resistance. And I do invite you to read the O'Neill report if you want to learn more about that. Now, a promising approach towards this goal is to study the natural ecological antagonists of bacteria in the gut, and that is bacteriophages. Now, studying the role of bacteriophages in the gut may illuminate new approaches to deliberately and rationally perturbing specific species, elucidating causality as mediated by interbacterial and bacterial mammalian host interactions, and ultimately designing precise and predictable approaches to remodel the gut microbiome for therapeutic purposes. Now, to address how bacteriophages impact bacterial communities in the gut, the researchers of this study investigated the dynamic effect of phages on a model microbiome. And how did they do that? Well, they used germ-free or neotobiotic mice, which were colonized with a defined human gut commensal bacteria consortia, and then subjected them to predation by cognate lytic phages. And the first step was to embark in the longitudinal characterization of phage behavior in the mammalian gut. And the model microbiota comprised of facultative and obligate anaerobic commensal bacterial species known to colonize the human gut and that can also co-colonize germ-free mice. The 10 selected species represent the 4 major phyla in the human gut microbiome, so the firmicutes, the bacteroidetes, the proteobacteria, and the verrucomicrobia. And you see these on the left in this panel. On the right, you see the phages that were used to target specifically some of these bacteria. And as you see in the image below, the experimental setup was very elegant and clear. In fact, individually housed mice, and there were 5 biological replicates, were orally gavaged with the defined bacterial mixture containing 2 × 10 to the 6 to 2 × 10 to the 7 colony-forming units per strain. And then on day 16 and day 30, mice were gavaged with sodium bicarbonate to neutralize gastric acids, followed by 2 × 10 to the 6 plaque-forming units of phage. The fecal samples of the mice were then collected periodically throughout the experiment, from which bacteria and phage were quantified using molecular methods such as qPCR and next-generation sequencing. Now, in this image, you can see the concentrations of each phage and targeted bacteria, respectively. The analysis revealed that both phages and their targeted commensal bacteria persist in the gut. In fact, after administration, each phage was detectable 4 to 6 hours after administration and continued to be detectable for the duration of the experiment. On the image in the right, what you see is the quantity of Enterococcus faecalis in selected samples of mice and the percentage of colonies that were found to be resistant to the lysis by the VD13 phage as determined in vitro. Now, these results suggest that phage-directed knockdown of bacteria leads to the enrichment of a phage-resistant subpopulation. And in fact, as you can see, just before and just after the administration of the phages, all colonies are susceptible to lysis, whereas as time progresses, there is an emergent population, subpopulation of bacteria, which slowly becomes resistant. So the next question that the researchers of this study addressed is, what is the effect of introducing phage on the commensal gut microbiota? And longitudinal tracking of each bacterial species revealed that phage predation induces quantitative shifts in the microbiota, including in those species not susceptible to phage. When examining the bacterial composition in terms of estimated concentrations for each species, as shown in this picture, they found that administration of phage induces shifts across the microbiota in both low and high abundant species. For example, after the first set of phage targeting E. coli and Clostridium sporogenes had been administered on day 16, they observed observable shifts occurring in both the low abundant species, such as Bacteroides vulgatus and Proteus mirabilis and Parabacteroides dissesonis, and the high abundant species, such as Akkermansia mucinifila and Bacteroides fragilis. Now, less obvious were the effects due to the second set of phage administration on day 30.1. But ultimately, the take-home message here is that phages induce cascading effects on microbiota species that are not directly targeted. Yes, and finally, in this slide, I think we can truly appreciate the value that metabolomics analysis run using Metabolon's global untargeted platform was able to bring to the study, and how it was really able to provide a lot of insight into the biotechnological potential of bacteriophages as microbiome modulators. In fact, next, the researchers sought to characterize the functional effects of phage predation on the microbiome as reflected in changes in the gut metabolome. And understandably, the expectation was that the levels of most metabolites would actually be buffered against fluctuation due to the metabolic redundancy across the defined consortia. But compounds associated with specific microbial pathways unique to particular species in the consortium would be sensitive to perturbations in bacterial composition. Now again, using Metabolon's untargeted HD4 Metabolomics platform, the researchers were able to survey the fecal metabolites during various stages of colonization in mice, namely in germ-free mice, after the stable bacterial colonization, then after the introduction of E. coli and C. sporogenous phages, and then after the introduction of E. faecalis and B. fragilis phages. Now, in the upper left-hand corner, you see that fecal samples were collected at different time points, mainly from germ-free mice colonized by the defined bacterial consortia, then after the treatment with the first set of phages, T4 and F1, and then after the second set of phages, VD13 and B48. And just below, in the volcano plots, you can see the increasing significance versus the fold change of each metabolite 13 days after administration of the first set of phages on the left, and then of each metabolite 13 days after the administration of the second set of phages on the right. And on the right-hand side of the slide, just to help in the direct comparison of how each metabolite changes relative to the bacterial consortia, you can see the hierarchical clustering of the mean fold change in bacterial concentration. Now, in this image, you can actually appreciate the output of the metabolomics analysis. In fact, this is the hierarchical clustering of significantly changing metabolites after introduction of the first set of phages in F, so most of the image, and then in the bottom right-hand corner, after the introduction of the second set of phages, which actually had a much more modest effect. And in general, I would say that overall, the phage-directed remodeling of the gut microbiota had a relatively modest quantitative impact on the metabolome. In fact, administration of the first set of phages resulted in statistically significant changes in 17% of examined compounds, but with metabolites interestingly representing nearly all KEGG pathways, so amino acids, peptides, carbohydrates, lipids, nucleotides, cofactors, vitamins, and even xenobiotics. Now, by comparison, the introduction of bacteria obviously into germ-free mice without phage treatment resulted in broad shifts in the fecal metabolome, enriching 60% and reducing 15% of the total 860 metabolites measured across all KEGG pathways. Now, taken together, these observations suggest that the breadth of metabolomic impact mirrors the extent of compositional shift in the gut microbiota. Now, most importantly, I guess, for this study, we were able to see how phages can modulate metabolites with known mammalian host effects associated with multiple bacterial species. For example, tryptamine, which is a neurotransmitter commonly of plant origin, but that can also be produced via a tryptophan decarboxylation, which can be conducted by bacteria, and in particular, Clostridium sporogenous, which was a member of the defined consortium. And during treatment with the first set of phages, we detected a 10, 17, and 2-fold reduction in tryptamine, 0.3, 2, and 13 days after treatment, respectively. Another example is the neurotransmitter tyramine, which is produced via the tyrosine decarboxylation by lactic acid bacteria, including E. faecalis, again, the sole lactic acid bacteria of our consortium. And administration of our second set of phages caused a 4, a 2.7, and a 4-fold decrease in tyramine, again, 0.3, 2, and 13 days, respectively, after the administration of the phages. Now, because of the limited catalog of experimentally verified microbial metabolites, it is difficult to broadly associate specific metabolites to individual species within our consortium. However, the unique associations of tryptamine and tyramine to Clostridium sporogenus and Enterococcus faecalis, respectively, clearly suggest clear causal links between phage, bacteria, and metabolite. Now, another example of compounds that were affected after the first treatment with phages were two amino acids, serine and threonine, which are highly represented amino acids in O-glycosylated intestinal mucin. And this is consistent with our observed enrichment of the mucin-degrading commensals Akkermansia mucinifila and B. vulgaris because of the phage effects. We also found significant changes in bile salts due to the first set of phage treatment. We found that the modulation of the bacterial consortium by the first set of phages increased the deconjugated bile salt cholate sulfate and decreased the conjugated bile salt taurochenodeoxycholic acid 7 sulfate. This suggests an increased activity of salt hydrolases, which we found prevalently associated with various members of the consortium. We also detected increases in two deconjugated secondary bile salts that were not detected in germ-free mice and thus microbially derived, and that is 12-dihydrocholate and ursocolate. Now, to conclude, in my opinion, this beautiful work really provides insight into the ecological importance of phages as modulators of bacterial colonization. And it additionally suggests the potential impact of gut phages on the mammalian host with implications for their therapeutic use or to precisely modulate the microbiome. But from the biotechnological perspective, I truly think that the insights that this study provides are polyvalent. For example, the same strategy could be used in applied markets, for example, in bioprocessing applications. Let's say, for example, in wine fermentation, if you want to modulate the organoleptic properties of a product like wine, or even in animal nutrition, to substitute the subtherapeutic use of antibiotics as performance or growth enhancers. Now, I truly hope this study was of interest to you, and please don't hesitate to contact Metabolon if you feel you require further information, or if you just want to begin discussing how our metabolomics platforms can help you overcome the challenges that you're currently facing in your research, therapeutic area, or product development. Thank you again for your attention. Ciao. And finally, this is a schematic summary slide of this absolutely beautiful study, which was eventually published in Cell, Host, and Microbe in 2019. First author was Brian B. Hsu. The title was Dynamic Modulation of the Gut Microbiota and Metabolon by Bacteriophages in a Mouse Model. And some of the main challenges that were faced in this study were to study the capacity of phages to impact the gut microbiome's composition, its functional output, and possibly to infer the impact on a mammalian host. Now, some of the insights brought by the metabolomics analysis revealed that the shifts in the microbiome caused by phage predation impact the gut metabolome, and that phages can modulate metabolites which are known to affect the mammalian host. And the findings of this research also led to enhanced understanding of the functional impact of bacterial communities in the gut through phages on a model microbiome. This study was able to highlight, I think, a new therapeutic avenue to modulate the gut microbiome. It provided mechanistic understanding of the ecological importance of phages as modulators of the gut microbiome, and it provided information about the potential impact of gut phages on the mammalian host microbiome and its capacity to modulate health. Thank you all again for your attention, and see you for the next case study. Ciao.